So in our discussion with Gregor Mendel and his P plants, based on the video we saw just a moment ago, we now know that the P generation had two parents, a tall parent and a short one. And we now know that the alleles that the tall parent had were both dominant alleles, and the alleles the short parent had were both recessive alleles. So we know that these parents were crossed to create the F1 generation, and all of the F1, they were all tall. So we're going to look at using a Punnett square to show how that came to be. A Punnett square, it kind of looks like a window. And when we fill out the Punnett square, which will show the probabilities of combinations of alleles for offspring, we have to put one of the two parents up on the top and the other parent on the side. Really doesn't matter which way you go. I'm going to put the tall one on the side. Now what's important to note is that we have to remember that this allele and this allele are on homologous chromosomes. And if we recall during meiosis, when gametes are made, homologous chromosomes are separated into different cells. They're no longer in the same cell. So a gamete will either have this chromosome or this chromosome, but not both. So it could be this chromosome, and if that's the case, then the child will get a dominant T. I'm going to put this one up here since we have to show the possible combinations of alleles if the child gets this chromosome. This one's also a big T. And I'm going to put that one down there. And what I am showing right here is essentially the law of segregation. The law of segregation states that these two homologous chromosomes, they have to separate. They have to segregate when forming gametes. And that's what I'm doing here. I'm showing these two alleles, they segregate. They are no longer together, they are separated. The same will hold true for these two chromosomes. I'll put this little T right here, and I'll put the other one right here. Now to complete this Punnett square really is now a multiplication table. Take this big T, bring it across into every box that's in the row that it's in. Same with this big T. I'll bring it across, and I'll bring it across. Now I've put this big T into every box in that row. Now I got to do the same with the little t's, but I got to bring them down. So a little T, so I'll bring it here, and I also need to put a little T here because it's in that row. Same with this little T, bring it down, and bring it down. Now what we have are the possible combination of alleles for the offspring between these two plants when they were crossed. And we can see a dominant allele, dominant, dominant, dominant. So it looks like 4 out of 4 offspring will have a dominant allele, which means they will be tall, which explains why the F1 was all tall. We now know that every single one of those offspring in the F1 also had one big and one recessive allele. But how about the F2? Keep in mind that some of the F2 were tall and some were short. So let's cross 2 of our F1 plants to get our F2. We know that all of the F1s are big T, little t, so both parents are going to be big T, little t. So big T, little t by big T, little t. Again, I have to segregate my alleles, separate them. I'll put the dominant one up on the top, the recessive one down on the bottom. And then for this one, that parent will go on top. Again, segregating the alleles. Bring this big T across into every box in that row. Do the same for this little t. Bring the big T down into every box beneath it. Bring the little t down into every box beneath it. And I have my F2. I can see that 25% will be big T, big T. I can see that 25, 25. So I can see that 50% will be big T, little t. And I can see that 25%, 1 out of 4, will be little t, little t. So that is showing me that 75%, or 3 out of 4, will be tall, and 1 out of 4 will be short. And that's what Mendel found. Tall, 75%, short, 25%. And he also found that of this 75%, 25% were big T, and 50% of the 75 were big T, little t, and all of the short ones were little t, little t. So we're kind of working through, we're now seeing how Punnett squares kind of are used to determine probable outcomes, not guaranteed outcomes. But there's a few more terms that we need to know before we move on. To start with, we'll go with our combinations of alleles. So big T, big T, big T, little t, little t, little t. These are known as genotypes. And genotypes is the combination of alleles for a gene. And we have gene and genotype for a reason. So I can see that there's 2 alleles here, both are dominant. There's 2 alleles here, both are recessive. There's 2 alleles here, one is dominant, one is recessive. So I have 3 genotypes based on my scenario above. And each of these genotypes is given a specific name. Big T, this one is called homozygous dominant. Homo meaning the same. Dominant because they're both the dominant allele. Similarly, the little t would be homozygous but recessive. So homozygous recessive. And again, homo meaning the same, little. And then we have the opposites. This genotype is called heterozygous. So we have 3 different genotypes: the homozygous dominant genotype, the homozygous recessive genotype, and the heterozygous genotype. So we have our 3 genotypes. We know that those 2 genotypes will create a tall plant. This genotype will create a short plant. And we know that those are the traits which, as you recall from an earlier slide, are also known as physical characteristics and are also known as phenotypes. Pheno, physical. So when we look at this, what's the relationship between the genotype and the phenotype? Well, this codes for short, those code for tall, What happens if I change one of those two alleles, or one of those, or one of those? Well, the phenotype might change. So we can say that genotypes, or the combination of alleles, they will ultimately determine the phenotype or the physical trait of an organism.